So next talk will be given by Jonah. I think it has been a slight change in the title, so the new title is Potential Influence of Lipid and Fatty Acid, Nutritional Status of Maturation and Spawning Time of Cod. So Jonah.
Thank you very much. I will tell you a bit about a study we have made on the potential influence of lipid and fatty acid nutritional status on the maturation and spawning time of cod. It's a study I have done together with Maria Røybæk, Josiane Støttrup, Charlotte Jacobsen, and Fritz Køster, all from the Technical University of Denmark. The original idea came from the change of Baltic cod spawning time. The spawning time changed during a period of regime shift in the Baltic Sea in the early 90s, which affected the recruitment and the stock dynamics. If we start with the lower figure down here, you can see that if we go back to the 70s, we had peak spawning time. The peak spawning time is measured from ichthyoplankton surveys, which are conducted during the season, and from a model, the peak spawning time is calculated. And you see during this period, we had spawning times which were in April and early May. But it changed with time, and when we arrive here in the 90s, yes, we are going all the way up to having a peak spawning in August. And if we compare over here, this is the same point as we have here, you can see that after this regime shift, it has remained relatively constant with a lower amplitude during this period. The reason why the spawning time changed has been focus of several studies. The ambient water temperature has been a factor studied. The stock demography has been studied, both whether it could be a matter of the age diversity, the stock being truncated, or it could be a matter of the size of not only age, but also the size of the fish. But none of these factors alone or in combination explains the shift in spawning time. But what actually happened during this period? What was the cause of the regime shift? The regime shift was climate-driven changes in salinity and temperature. The Baltic Sea is a water body which is brackish and only connected with the North Sea saline water through the Danish Straits. In periods which are dominated by western weather, there may not be very many inflows, and that affect both the salinity, the oxygen level, and the higher temperatures. So the cod is living in the deep saline water, and the sprat and herring, which are their main prey, are more frequently also using the other more freshwater layers. The change, which went in direction of lower salinity of the water layers and higher temperatures, caused a food web change in the whole ecosystem. So when we go back in time, we will see that the phytoplankton in spring, the phytoplankton bloom, was dominated by dinoflagellates, but later it changed into diatoms. We have that the zooplankton in spring was in former time dominated by Pseudocalanus ascuspes, but it changed with the regime shift to Arcadia species. If we look at what happened with the cod and sprat populations, then you see that the sprat population that were low at this time, also being preyed upon by the cod, increased tremendously in number, while the cod population decreased over a very short time period. What happens also during such a regime shift is that the food web production of polyunsaturated fatty acids, the PUFAs, change. So the dinoflagellates, they are producing different lipids than the diatoms. And when we see here the polyunsaturated fatty acids, they will go from the phytoplankton into the zooplankton. The zooplankton will go into the planktivorous fishes, which are then eaten, by the cod. I think now I have to try to find my way back. And what happens then in the system is that the cod and also the clupeids They are not able to synthesize these PUFAs themselves. They need to get them from the phytoplankton, and consequently, there may actually arrive a limitation of PUFAs in the system with this regime change. So our hypothesis was that the altered spawning time and reproduction success could be caused by lipid energy storage due to changed diet composition, or it could be due to deficiencies of essential fatty acids, in particular PUFAs, important for fish reproduction, in particular arachidonic acid, EPA, DHA, or the mutual ratios. We could not compare with the former times because such analysis had not been made before the regime shift. So we decided to compare Baltic cod with North Sea female cod in the late maturation state to see whether the accumulated levels of lipid energy in ovaries and liver differed between cod in the two areas. And we made an analysis of the fatty acid composition in specific maturation stages. So we looked at the fatty acids changes and the composition during female cod maturation in the ovaries and in the liver. And we investigated the fatty acid composition in both ovaries and livers in cod in late maturation from the two areas, comparing in particular ARA, EPA, and DHA. This is our sampling area. We sampled in the Baltic Sea down here, and we sampled in the North Sea, where we got help also for Peter Wright and the Marine Laboratory in Aberdeen. It was all sampled on board on surveys. The fish length, weight, organ weight were measured, and from the ovary we took a small sample for accurate determination of maturity histologically, and we took a sample of the liver for lipid analysis in the lab. So the histological processing, we divided the fish into immature and resting, both characterized by primary growth oocytes, as we have here. We are having the early maturation stages. We divided them into 3 substages. So we have the cortical alveoli, M30, we have M31, which is the early vitellogenesis, and we have M32, which is the mid-vitellogenesis. And then we have the stage 4, dominated by fully yolk oocytes. The lipid analysis was conducted on ovaries and liver, and we used FAME analysis and gas chromatography to look at the lipid or the fatty acid composition, and we separated between phospholipids and neutral lipids. Here you can see we had in total 213 female cod, 198 from the Baltic and 25 from the North Sea. The first results we got was looking at the GSI and the ovary lipid and the hepatosomatic index and the liver lipids. If we look here at the 3 columns to the left, that is the Baltic cod, those ones which are not maturing, they are either close to being mature or they are still late immature, and we have those ones that are resting or skipping spawning. Then we have the maturing ones and the late maturation. There is a clear increase, a significant increase in the GSI, and when we compare to the North Sea cod, the North Sea cod are having larger GSIs on average. That has also something to do with the size of the fish in our study. If we look at the ovary lipid, we see that if we compare the North Sea and the Baltic Sea, there is no significant difference in the ovary lipid level between the two, while it is building up in the Baltic Sea. If we go to the hepatosomatic index, we do not see very much difference when we look at the hepatosomatic index, except we see that it is significantly higher in stage 4 in the Baltic compared with the other ones. But if we look at the liver lipid, it actually gives a much more detailed impression and even actually changes the order of the different maturity stages. What is in particular interesting is that we have here, if we compare the North Sea cod and the Baltic, the Baltic cod and the North Sea cod, we see that there is a significant difference. The lipid level is much higher in the Baltic Sea than in the North Sea. So it has no reason to believe that they would be limited in lipid energy from their food sources. When we looked at the fatty acid composition, we looked at the ovaries. We have used PCA analysis, where we included morphometric data around 35 fatty acids, maturity, lipid, and so on, to look at the development. I actually wanted to go back, but I can do this. If we look at how the different ovaries in different maturity stages they distribute in this loading plot, you can see that here we are having the ovaries which were not very developed, the either skip of spawning resting or late immature, they are in this quadrant. Then we have the maturing ones, which are in this area, the early maturity, and we are having here those ones that are in the late maturation stage. When we look at the late maturation stage, we can see that they are related to a high lipid level, to high liver condition, to high level of PUFAs, while the EPA arachidonic acid level is high, which also tells that there's a negative correlation between the late maturation state and the arachidonic acid level. And that was particularly this fatty acid that we thought might actually be limiting in the Baltic Sea. It is relatively higher in those ones that have not started the maturation. If we look at the two different areas, we find significant differences in the phospholipid composition among the ovaries in the late maturation stages. You recall there were no difference in the total lipid in the two in ovaries from the two different stocks, but when we look at the fatty acid composition, there are significant differences, and we are seeing also when we look at the North Sea that there seem to be two groups depending on whether they are from the southern North Sea or the northern North Sea. The corresponding loading plot is indicating how the development or how the relationship is between the different kind of fatty acids in the Baltic Sea and in the North Sea. So in the Baltic Sea, we can see we had a very high liver index, we have high level of the PUFAs, we have lipids, and we have a high level of DHA, but a negative correlation to arachidonic acid, while the North Sea cod ovaries show a positive association with ARA. This confirms what also is known from other studies, that ARA is very important in the maturing ovaries. If we look at the livers, we have a loading plot which is showing how it looks like in the North Sea. And here we can see that the liver of the North Sea cod is dominated by MUFA, the monounsaturated fatty acids, and the saturated fatty acids, whereas when we see in the Baltic Sea, they have both the very high liver index, have high liver lipid, and they also have very high levels of both DHA and EPA, while the ARA is low. There is a positive correlation between the DHA and the GSI in the ovaries. That is shown also here, where we can see that in the maturity stages, the early maturity stages, the level is relatively low, but it seems that it is selectively accumulated in the ovaries, and when we reach the level where we are having the high GSI levels, the level of the DHA is also high. DHA is a very important phospholipid in the membranes, and it has a high importance for the quality of the eggs through the membrane structure and function. There was a negative correlation between arachidonic acid and the GSI. You can see here how the arachidonic acid is relatively high in the early stages, while it is falling towards the when the GSI is increasing, and the arachidonic acid is a important precursor of many sex hormones, and they are very important for both the onset of maturation and also the early maturation period. If I should summarize the differences in lipid composition between the late maturation ovaries between the two areas is mainly that the hepatosomatic index and the level of stored liver lipid was significantly higher in the Baltic cod than in the North Sea cod. So they stored more energy, but could not use it for reproduction. The proportion of DHA was significantly higher in the Baltic cod than in the North Sea cod, and the proportion of ARA and EPA was significantly lower. If we look at the differences in lipid composition among the Baltic cod ovaries in different maturity stages, it was in particular the proportion of DHA that increased with progressive oocyte maturation and the ovarian development during the maturation stages to the late maturity ovaries. The proportion of the ARA decreased in the ovaries with progressive ovarian development through the maturity stages we looked at. And if we conclude in relation to the hypothesis we set up at the beginning, we found that the high level of liver lipid in Eastern Baltic cod ruled out that the poor energy reserve could be a cause of the change in the spawning time of Baltic cod. What we found was that the low level of the N6 essential fatty acid arachidonic ARA in Baltic cod ovaries and livers would support that ARA could be a limiting factor in ovarian maturation and reproduction of Baltic cod. Thus, the changes in the food web affecting clupid prey quality may cause a diet of Baltic cod rich in the N3 essential fatty acids and SFAs, but insufficient in the essential fatty acid arachidonic acid. So what's next? What's next would be to resolve more of the food web, the seasonal lipid and fatty acid dynamic in cod in other stages than those ones we were looking at, and to see how it actually looks like in the clupids. Are they also low in arachidonic acid? They are the main prey of cod, and that would be of value to know whether it is also the case for the clupids. Another issue would be to do experimental work to clarify the influence of different dietary levels of arachidonic acid, EPA, and DHA on cod reproduction in general. That is the next presentation that will deal with these things. I will say thank you for your attention.